Okay, the slide is visible. My dear friends, the slide is visible. Okay, shall we start this class, my dear friends? Anjana madam garu.
Yes sir, yes sir.
Okay, start చేసుకుందాం. రికార్డ్ స్టార్ట్ చేయండి.
చేశాను సార్.
Okay. Okay, very good evening, my dear friends. Welcome to the session. So, yesterday class we have discussed about the nitrogen metabolism. In that nitrogen metabolism, we are clearly discussed about nitrogen cycle. So, how many process, how many ways of nitrogen fixation? Maybe the atmospheric nitrogen fixation. Also, the second one is the industrial purpose nitrogen fixation, and the third one is the most important, that is biological nitrogen fixation. So, it is three ways, three types of the nitrogen fixation. So, one of the important area and most useful topic today we will going to discuss today's class, that is the biological nitrogen fixation. So, fixation of the nitrogen through the physical mode or chemical mode or the microorganisms which they are involved in this process. Okay, so in this process, microorganisms, mainly the microorganisms which they are belongs to prokaryotes, especially bacteria and cyanobacteria like members, even though some of the eukaryotic members like fungal members also involved. Fungal members also involved in this biological nitrogen fixation. So, in this biological nitrogen fixation, so nitrogen, the molecular nitrogen which is present in the gaseous state in atmosphere, highest largest amount of the nitrogen gas, that is 78%, which they are present in atmosphere. Am I right? Are you agree or not? How much percentage?
Yes sir.
How much percentage present in atmosphere nitrogen? 78. Very good. 78% of the nitrogen present in atmosphere, it is a gaseous substance. It is a molecular state. It is in molecular state. So, this dinitrogen gaseous substance is unable to utilize by all higher plants. So, the unusable forms is converted into usable forms by the help of these microorganisms. The microorganisms may be the bacteria, the microorganisms may be the cyanobacteria, the microorganisms may be the fungal members. So these living organisms which they are involved in conversion of the atmospheric nitrogen into usable forms like ammonia, whatever it may be, nitrates and nitrates. These are the forms, usable forms. So these usable forms is converted with the help of living organisms. So the biological being or biological involvement, the dinitrogen, which is a gaseous substance which is present in atmosphere, that is converted into usable forms. So this nitrogen fixation we can call as biological nitrogen fixation. And my dear friends, remember here, the availability of the nitrogen, which is a limiting factor of the all plant growth. Why? Because the dinitrogen is not directly used. The plants, they cannot directly use. Just they are used in the form of other forms of nitrogen like ammonia, nitrates, and nitrates. Even though ammonium ion also. So the ability of the fixation of the living organisms, they are mainly prokaryotes. Okay, so they are bacteria. So the bacteria, maybe the symbiotic bacteria, maybe they are not associated with any other living organism. They are free forms. They are lived outside the environment. So the bacteria which they are free living bacteria, or the bacteria may be the symbiotic bacteria. They are associated with higher plants. They are associated with higher plants. So these bacteria can fixed from atmosphere. Fixed from atmosphere. So the first attempt to increase forest growth through nitrogen fixation in Lithuania in 1894. So especially lupines in Scots and pines. Okay. So in this biological nitrogen fixation, some amount of the energy is also necessary, even though some electrons and protons are. The slide is visible. Biological nitrogen fixation. The chemical equation is visible, my dear friends. The chemical equation is visible. Is the chemical equation?
Yes, sir. To admit.
Huh?
Somebody are waiting to admit, sir.
అమ్మా నాకు వినపడలేదు మీరు చెప్పింది. వెయిటింగ్
అడ్మిట్ చేయరా సార్? మా ఫ్రెండ్స్ వెయిట్ చేస్తున్నారు.
మరి ఎవరు చేయాలి? మేడం ఉన్నారు కదా, మేడం చెప్పాలి.
ఓకే జాయిన్డ్ మౌనిక.
ఓకే, ఇప్పుడు ఈ సెకండ్ స్లైడ్ కనబడుతుందమ్మా?
ఎస్ సార్.
ఓకే, ఈ కెమికల్ ఈక్వేషన్ ఉంది ఇక్కడ చూడండి. సో ఇన్ బయాలజికల్ నైట్రోజన్ ఫిక్సేషన్, the all microbes like bacteria, cyanobacteria and other fungal members also involved in this biological nitrogen fixation. A special type of mechanism is also involved in this process. The mechanism is nitrogenase enzyme. So nitrogenase is an enzyme which is involved in the biological nitrogen fixation. In this biological nitrogen fixation, nitrogenase is contained a special type of what we can say that nutrients or special type of minerals, that is molybdenum and ferrous. So molybdenum, ferrous units are present in nitrogenase enzyme. Along with this nitrogenase enzyme are the genes which they are control the nitrogenase activity. The genes we can call as nitrogen fixation genes or NIF genes. What we can say that? NIF genes. Okay, here in this slide we are observed here. So nitrogen. This is the molecular nitrogen or dinitrogen, and 8 flavonoxin. 8 flavonoxins are necessary. At the same time, 8 protons are necessary. Along with this, how many ATPs are necessary for this biological nitrogen fixation? 16 ATPs. How many ATPs are necessary? 16 ATPs are necessary. 8 protons and 16 ATPs are necessary. Very, very important. Even though competitive examination also, this how many ATPs are involved in this process. At the same time, how many protons are involved in this biological nitrogen fixation? Come on, tell me how many protons are involved? How many protons and how many ATPs are involved? Come on.
16 ATPs and 8 protons.
ఒకటి ఆన్లైన్ క్లాస్ లో ఉన్నా. డెఫినెట్లీ అన్న, అరే మీరు చెప్పిన తర్వాత వాళ్ళ తిరుగులేదు. అన్న ఇట్లా అట్లా ఫాలో అయిపోదాం. ఓకే, షూర్. రైట్, థ్యాంక్ యూ. రైట్. So in this process, my dear friends, 8 protons are necessary. At the same time, 16 ATPs are mandatory for fixation of the nitrogen from atmosphere in the biological nitrogen fixation. So 18 ATPs, very, very important. At the same time, this nitrogenase enzyme is also, along with this nitrogenase enzyme, magnesium ion is also involved or utilization in this process. So then, dinitrogen, 8 flavonoxin and 8 protons and 16 ATPs and 18 H2O molecules. H2O molecules means 18 water molecules are involved in this process. So this nitrogenase enzyme it is converted into, see here, so dinitrogen or molecular nitrogen is converted into two ammonium ions. Two ammonium ions. Okay, two hydroxyl ions, 8 fluoroxyl. Let's see here 16 ADP. So 16 ATPs are involved, utilized, and 16 ADPs are here, and phosphate. So phosphoric acid or phosphate 16 H2PO4 is released. See, ultimately, the what is the final product? End product here, two ammonium ions. So one dinitrogen molecule is converted into how many ammonium ions? Two ammonium ions is released, or two ammonia atoms are released. So this mechanism is we are observed in microbes like bacteria, cyanobacteria, even the fungal members also. This is internal mechanism. This chemical equation completely explains how dinitrogen of atmospheric dinitrogen is converted into ammonia or ammonium ions. Okay, see here. So this rare and extremely energy consuming conversion because of stability of triple bonded end. See here, my dear friends. Here, I'm unable to write here. So in this process, so in between two nitrogen atoms, triple hydrogen bonds are strictly binded. So these triple bonds is diluted or converted or breakdown by this nitrogenase enzyme. So in this process, 16 ATPs are required. How many ATPs are required? 16 ATPs are required. So it is energy consuming process. Are you agree or not? So this dinitrogen or molecular state of nitrogen is going to break down between two nitrogen atoms. Triple hydrogen bonds is break down by the utilization of 16 ATPs energy. Am I right or not? So in this process, this produces nitrogen which can be directly assimilated into nitrogen containing biomolecules. That is ammonia. Okay, here the third slide, I hope you clearly visible. ఈ స్లైడ్ కనబడుతుందా? ఈ మూడో స్లైడ్ కనబడుతుందా?
శనివారం పండుగ.
Yes, sir.
Okay, right. So in this image, we are explain. So what you observed, come on, what you observed in this slide, clearly explained. Very with our naked eye, we are easily understood this image.
ఎందుకంటే శుక్రవారం నమాజ్ అవుతున్నది.
మళ్ళీ పెళ్ళికి ఏమైనా అవసరం ఉంటే తీసుకోవాల్సి వస్తుంది కదా, మళ్ళీ పెళ్ళప్పుడు కూడా అవసరం ఉంటే తీసుకొస్తే.
Okay, see here in this slide, see here dinitrogen molecule is converted into ammonia. So this is the nitrogen fixation process. In this process, whether the bacteria they are associated with symbiotic association associated with the legume plants, or some of the azospirillum bacteria which they are associated with non-legume plants, and also some of the bacteria they are lived in free living manner. They are lived in the soil. Am I right? So the best example of symbiosis, the bacteria which they are associated with the root nodules. See here, so Rhizobium leguminosarum, and see here the second image. This is the azospirillum, and this is rhodospirillum. Am I right? See here, so energy source of organic carbon. Energy source of organic carbon, it is absorbed from host plant here, and here root exudates from the host plant. But here heterotrophs, plant residues are autotroph photosynthesis. If maybe the bacteria, for example, rhodospirillum, so that is photoautotrophs, you know. So then automatically the energy source is organic carbon, the energy source which is absorbed from photosynthesis. Okay, and if they are belongs to heterotrophs, heterotrophs. They are absorbed from plant residues. Whenever the dead decaying material like dry leaves, they are going to wet and decompose in the soil. Then the plant residue can utilize the organic carbon energy source. This organic carbon is plant residue. Okay, so then in this process, the estimation of the fixation rate of the nitrogen kg for hectare, kg for hectare in year. See here, so symbiosis manner 50 to 400 kg. Then association with they are non-leguminous association, they are 100 10 to 200 kg per hectare. And here, whether they are free living bacteria or they are heterotrophs, 1 to 2 kg per hectare per annum. At the same time, whether they are autotrophs, maybe the photosynthesis bacteria, they are fixation of the nitrogen from atmosphere into how much kg? Come on, 10 to 80 kg. 10 to 80 kg. 10 to 80 kg of 10 to 80 kg of nitrogen fixed by these three living bacteria. So symbiosis bacteria can ability to fixation of the nitrogen from atmosphere high amount of nitrogen. Are you agree or not? See here, 50 to 400 kg. But they are associated with non-leguminous plants, roots. They are fixation of the nitrogen from atmosphere 10 to 200 kg per hectare. But whereas in free living bacteria, very little amount of nitrogen is fixed, 1 to 2 kg and 10 to 80 kg per hectare. Am I right? No.
Yes.
You are not. Am I right? Is it right or not?
Yes, sir. Yes, sir.
So, this image is shows about the, how different bacteria they are involved in this nitrogen fixation process. And my dear friends, remember here, this biological nitrogen fixation is also we can call as diazotrophy. What we can say that? Come on, tell me anybody. Diazotrophy. The method of nitrogen fixation we can call as diazotrophy. Which method? The biological being like microorganisms, they are involved in the nitrogen fixation process. The entire process we can call as diazotrophy. Diazotrophy. Di means two. Di means two. Azo means nitrogen. Trophy means living organisms. So the living organisms like bacteria, cyanobacteria, or some of the algal members, they are involved in the fixation of the nitrogen from atmosphere. That entire process we can call as diazotrophy. This diazotrophy has first clearly explained by Jagtial. Who discovered? Jagtial.
Okay, but Korea.
Which scientist?
But Korea.
My dear friends. Which scientist? Jagtial. Okay. Some here, see here. So some of the bacteria, maybe they are heterotrophs, maybe they are photoautotrophs. If they are heterotrophs, maybe they are free living bacteria, symbiotic bacteria. Okay. If they are photoautotrophs, maybe the free living bacteria, symbiotic bacteria. If they are symbiotic bacteria, they are associated with legume plants. They are associated with non-legume plants. If they are photoautotrophs, even though they are symbiotic, they are non-nodule forming bacteria and nodular forming bacteria. Okay, so these are this table this flow chart is clearly explained how diazotrophy involved in the nitrogen fixation. Maybe the formation of fruit nodules, maybe they are free living associated with other plant parts, maybe the roots or other internal parts of the plants. Okay, so here the nitrogen fixation requires energy input. See here, my dear friends. So in this process, reduction reaction, so electrons must be added. Just because, why? Because sensitive to oxygen. My dear friends, remember this nitrogenase enzyme, very very important. Nitrogenase enzyme is a very important enzyme, which is also very sensitive enzyme sensitive to oxygen. Okay, and it requires 13 kilojoules of energy per mole of nitrogen fixed. It is theoretically the actual cost of what we can say that 15 to 30 grams per gram of ammonia produced. Okay, then the assimilation, the final assimilation of ammonia into organic forms takes 3.1 to 3.6 gram per the total nitrogen fixation. See here. Then in this process, nitrogenase enzyme is involved. Am I right? Nitrogenase enzyme. Okay. So in prokaryotic living organism like bacteria and cyanobacteria, so rhizobia fix nitrogen in symbiotic association with leguminous plants. Are you ever heard of leguminous plants, my dear friends? Give any examples, one or two examples. Very quickly. Very quickly. Remind immediately. Okay. Very good. So all pulses, they are belongs to leguminous plants. You know, Bengal gram, green gram, groundnut. Okay, soya bean. So they are belongs to leguminous plants. So these leguminous plants associated with Rhizobium leguminosarum bacteria. Okay, so these rhizobia fix nitrogen for the plant, and plant provide rhizobia with carbon substrates. Okay, carbon substrates. Then all nitrogen fixing system appear to be identical. So all are identical. Here they require nitrogenase enzyme. They are along with nitrogenase enzyme. My dear friends, remember here, reductant also involved. Reductant means reduced ferridoxin. I'm already told that this nitrogenase enzyme is involved in the nitrogen fixation process. This nitrogenase enzyme is contained two specific components, that is the iron or molybdenum, that is the ferrous molybdenum or iron molybdenum segments are present in nitrogenase enzyme along with this ATP. And even though oxygen-free conditions are most important. So if oxygen is present, the nitrogenase enzyme is inactive position. Without oxygen, nitrogenase enzyme is very, very active. If oxygen is present, then this nitrogenase enzyme is very sensitive to oxygen. So that is malfunction or not function properly. So oxygen-free conditions in environment is mandatory in this. Of NIF genes. NIF genes means nitrogen fixation genes. What is NIF genes, my dear friends? Nitrogen fixation genes. ఇది కనబడుతుందా మీకు? ప్రొకారియోట్స్ లో జరిగే మెకానిజం గురించి చెప్తున్నాను. కనబడుతుందా మీకు ఇది?
Yes, so yeah.
Outside.
Okay, it is visible. The slide is visible.
Yes, sir.
Okay, so the enzyme, so oxygen sensitive enzyme, that is nitrogenase enzyme. Very, very important. Nitrogenase enzyme very sensitive to oxygen. If oxygen is present, nitrogen is in inactive position. If oxygen is not present or anaerobic condition is there, then automatically nitrogenase is in active position. Okay, active position. But whereas, see here. But in aerobic condition means oxygen presence. So if oxygen is present, nitrogen is not properly working or inactive position. Remember, my dear friends. Okay. At the same time, the reducing power is produced by NADPH, that is via an iron molybdenum center. Iron molybdenum center. MO means molybdenum. Fe means iron. Okay. At the same time, plant genomes lack any genes coding this enzyme, which occurs only in prokaryotes like bacteria, cyanobacteria. If even within a bacteria, only certain free living bacteria like Klebsiella, Azospirillum, Azotobacter, they are in free living bacteria. But some of the bacteria, they are associated with legume plants like Rhizobium leguminosum is there. But here, what we discussed, Klebsiella, Azospirillum, and Azotobacter, these are all free living bacteria. Along with this, some of the blue green algae like Anabina, Oscillatoria, okay, nostoc, and a few symbiotic rhizobial species are known nitrogen fixers. So these are all nitrogen fixers. Nitrogen fixers means the living organisms, the microbes, which they can ability to fix the nitrogen from atmosphere. Are you agree or not? Are you follow or not, my dear friends? Respond quickly. స్పీడ్ అవుతుందా స్లో చెప్తున్నాను? స్పీడ్ అవుతుందా స్లో చెప్తున్నానా?
నేను రీఛార్జ్ చేయించలేదు.
అయ్యో ఎట్లా చేసిన
Nobody can respond.
పెద్ద పెద్ద
Okay na?
200 పెట్టాను.
వింటున్నారా?
ఓకే సార్.
ఓకేనా? బయట పోతే
వింటున్నారా?
గూగుల్ పే కాకపోతే నిమిషం పనికి వస్తే 400.
One side కాదు, two side మన involvement ఉండాలి, my dear friends. Remember, the another nitrogen fixing association exists between an actinomycetes, that is the Frankia species. Actinomycetes species is a special type of bacteria. Actinomycetes is a special type of bacteria which are very related to or very nearer to very closer to fungal members. So that bacteria we can call actinomycetes bacteria. The actinomycetes bacteria best example is Frankia species and also Alnus species. So these species are non-leguminous plants which they are produce the root nodules in their roots.
ఎక్కడిది
Okay. Frankia and Alnus. And the another important slide. This slide is visible. The enzyme nitrogenase catalyzes the conversion of atmospheric gas into ammonia. See here. So this is nitrogen and this is oxygen. So nitrogen and oxygen both they are associated to produce 2 moles of ammonia. Here, this is visible. 2 moles of ammonia is visible.
నేను అంతా చూస్తా అయ్యో
So the reaction seen simply enough in the atmospheric nitrogen, how much percentage of nitrogen is present in atmosphere? How many? 78% of the nitrogen is present in the atmosphere. Highest amount of the gaseous substance, highest amount of the elements which are present in atmosphere, that is nitrogen. Okay. Then some of the friends is asking me personally, sir, why this enzyme is so important? This enzyme means nitrogenase enzyme. This enzyme means nitrogenase enzyme. So the strong means the triple bond of nitrogen makes this reaction very difficult to carry out efficiently. So the efficiently the nitrogen or molecular state of the nitrogen, then between these two nitrogen atoms, the triple nitrogen hydrogen bonds or triple bonds are there. So the nitrogenase enzyme contains 16 moles of the ATP. ATP means energy, energy molecules. So these energy is lies and breakdown or
ఫోన్ తీసుకొని ఒకసారి
or nitrogen
ఏ కన్నయ్య
into ammonia. So
ఫోన్
which
ఇందులోనే
makes
రావు
one
ఇవన్నీ
of the
ఫ్లాష్
most
లైట్
energy expensive.
దీనికి ముందు వచ్చింది రావట్లేనా చూద్దాం పెట్టు
And what is nitrogenase complex? We know that nitrogenase is a complex enzyme, so which contains either molybdenum or ferrous molybdenum substances. You know,
ఏ కదలకురా
and my dear friends, remember here nitrogenase complex is two
అదే రోజు
components.
కూర్చుంటారు అంటాడు.
That is the nitrogenase
దగ్గరికి
and nitrogenase. See here, so nitrogenase is a 60 kiloton
ఇంటికైతే
homodimer with a single
నవ్వుతాడు కదా
ferrous sulfur cluster. My dear friends, remember, so it is the nitrogenase teenage reductation
కొట్టినట్టుంది.
with a single iron and sulfur cluster is present. Whereas, and remember here, my dear friends, nitrogenase is an enzyme which
అయ్యో
is
వీడు
very
హోంవర్కే
sensitive
రాయలేదు.
to oxygen molecule. And the nitrogenase enzyme binds with magnesium ion along with ATP adenosine triphosphate. And 4 ATP is required per pair of electrons transfer. How many ATPs? 4 ATPs. Then 4 ATP means in our equation how many ATPs are required? 16 ATPs are required. So then how many pairs of electron transferred? 4 pairs of electron transfer. Am I right or not? So in this process, the reduction of nitrogen to ammonia,
అమ్మ బర్త్డేకి కేక్ ఎందుకు కట్
protons or
చేస్తారమ్మా?
hydrogen requires 4 pairs of
Very
electrons.
good, just
So
a
16
ఏ
molecules
చెప్పు?
of ATPs are
కేక్
consumed
బదులు
for
వేలు కట్ చేస్తే అయిపోతుంది.
1 mole of nitrogen or dinitrogen. See here. So this is a equation, chemical equation. You know, 16 electrons,
Yeah,
and 16 protons. So
చాచి
this biochemical energy required here. The triple bond present in between two nitrogen atoms. How many bonds are present?
నొప్పి
Triple
తగ్గుతుంది.
bonds.
తీయకుండా
See,
But
these
finish
triple bonds is going to
కూర్చుకుంటే
cut down,
Kuchkodu.
break down by
Melloga ayinde
nitrogenase enzyme by the uses of the
సెట్కి అయితే
16 ATPs of amount of energy. Then it is converted into the end product or the final product, that is 2 mole of ammonia. 2 mole of, so 1 mole of dinitrogen is going to convert it into 2 mole of ammonia. So in this process, 16 ATP
ఒకటి
energy.
ఇచ్చిన వాళ్ళ
How much
opinion.
ATP? 16 ATP energy is required. So it is a reduction process, my dear friends. Reduction process. So nitrogen reduction to ammonia
వీడు ఏం
is
చేస్తుంది?
thermodynamically favorable. Thermodynamically favorable. And here, however, the activation barrier of breaking the triple bond between the two nitrogen atoms is enormous.
అమ్మ ప్రియా మీనాకి
Utilize
చేయకు.
16 ATP amount of energy. Okay.
బ్లాక్ డల్.
Then in this nitrogenase enzyme,
సీత
220 kilodalton. You know,
కట్టు హ్యాపీ
and
నేను అక్క
in previous
ఆహా
slide,
నాకు నాకంటే
so
నువ్వు
the
క్లాస్ పెద్ద కదా
nitrogenase
ఏమవుతుంది
complex,
చెప్పు
is an enzyme, it is a
చెప్పు.
protein,
ఏం కాదు
so which contains 16 kilodalton
నేను
homodimer.
చేసినప్పుడు రా అంట
With a single
అనొచ్చు
iron
2 క్లాసెస్ గా నాకన్నా
cluster.
చేసి
But
4
whereas
నేను అంటాడు అనొచ్చు కదా నేను అంట
the nitrogenase enzyme
3-1
is a 220
Meenakshi,
kilodalton heterotetramer.
Anand.
That is homodimer, nitrogenase
KR
enzyme.
KR
నగరమా?
Homodimer.
కరీంనగర్ కరీంనగర్.
Okay, but here the
అమ్మ మీనాక్షి వాళ్ళు
nitrogenase enzyme 220 kilodalton at the same time
Kandera.
heterotetramer.
అందుకే కండేరా
Heterotetramer. Here
ఏమిది
the
పిన్నిస్తే అంటే?
nitrogenase
కండేరా అంటే ఏంటిది
contains
ఇంటి పేరు ప్రియరబాటు
contains 4 moles of iron, sulfur
ఆ కొత్త
substance only. But whereas nitrogenase enzyme contains 2 molybdenum,
మంచిగా పడితే
and
పెట్టి
32 ferrous, and 30 equivalents of acid labile sulfide. That is FeS clusters.
కట్ చేసి
These are present in
నేనే చేసిన మీకు నచ్చదు ఏం
and
నచ్చదు
at the same
అది
time,
టక్కన
sorry,
ఎత్తుతుంది
nitrogenase
పనులు ఉంది కనుక
at the same time,
ఎత్తలేదు
4 iron
అదంతే
and 4 sulfur clusters plus 2 ferrous, molybdenum, cobalt,
గుండె ఇప్పుడు మనకి
and
కొమ్ములు వస్తాయి
iron molybdenum cofactor is present in this nitrogenase agent. So nitrogenase is a slow 12 electron pairs per second. At the same time, only 3 molecules of nitrogen per second.
50-40
So
ఇక్కడ
this
ఒక
is the
బాక్స్
threading cluster. We
తమ్ముని
need not to
బుక్లో రాయి వాడు
tell
రావు.
about
No,
this.
no, no,
These
ఏం
end
చేయాలి?
products. See here, this is how
ఇలా
iron, sulfur, and molybdenum electron transfer cofactor is binds here.
నాకు కూడా
How many types of the nitrogen
మంచిగా
fixers
ఉంది,
important in forest soils?
నేను చెప్పు
Forest soils.
birthday
How many
రాలే.
nitrogen fixers
నా
see here?
బుక్
These are three. That is the
When
cyanobacterium,
you-
heterotrophic
నా
bacteria,
బర్త్డే
and symbiotic bacteria. So cyanobacteria
మరి
may be
birthday
the nostrils, may be
మరి
the
ఎప్పుడు
anamina, may be
కావాలి?
the oscillatoria species.
అన్నయ్య
They
birthday
are
కావాలా?
associated with
కొత్త సైకిల్ కొన్నదా
other living organs, other
అట్లయితే
living
నాకు
plants.
డింగచిక ఒకళ్ళు కరీంనగర్ సైకిల్ తప్పితే కుక్కలు భయపడకుండా పోతా
Bacteria,
డాగుని
even though
భయపడకుండా
living
పోతా
bacteria
సైకిల్
are
నుంచి
associated with
ఇంత స్పీడ్
rhizospheres.
కావాలంటే
Rhizospheres means
ఓకే
area or part of the roots.
ఏమో.
They use energy from decomposing
నేను
organic matter to fix nitrogen, protect nitrogen by rapidly converting oxygen to
అమ్మ
carbon
దగ్గర
dioxide
తిరిగి
through respiration.
ఏం
Then
రాదు
the
దీంతో
third
ఒట్టి
types
తొక్కడం
of the
తప్ప
nitrogen fixes, they are symbiotic bacteria
చేతిలో
like
తొక్కడం
rhizobium, bradyrhizobium.
cycle.
Okay, for legumes and frankia, for non-legumes, even though the nodules contains a form of hemoglobin which binds oxygen and protects nitrogen. So what we discussed in previous slide, nitrogenase enzyme is oxygen sensitive. So hemoglobin, that is the
లాస్ట్ పిన్ని సిస్టర్ పని
is
చేసి
acted as an oxygen scavenger. Remember, my dear friends, oxygen scavenger is
మీనాక్షి
hemoglobin.
మీరు ఏం పెడతావు?
మీనాక్షి
So liquid
Happy
plant
birthday,
which
Meenakshi.
they are released, that red colored,
హ్యాపీ బర్త్డే
the
మీనాక్షి
red
అక్క
pigment
అబ్బా.
చెల్లె
area we can call as leg hemoglobin,
Happy
which
birthday,
is red
Chella.
color, which binds oxygen and acted as a oxygen scavenger. So
హ్యాపీ బర్త్డే మీనాక్షి
mechanism
Tata.
leads to the very actively involving conversion of the atmospheric nitrogen to usable form of the ammonia nitrates of this nitrogen.
హ్యాపీ బర్త్డే
See here.
మీనాక్షి
So the
Pichara.
purification
ఇప్పుడు
clepsydra.
పోయింది. ఏ?
ఓకే
Clepsydra. So
Yes.
this system is there now.
ఆ, సరే, ఆగు.
Started on when no fixed nitrogen.
చూడవా,
It is
ఇది ఒకటి.
mandatory anaerobic condition, and even though the temperature below 30 degrees Celsius, temperature below 30 degrees Celsius, and even though the
హ్యాపీ
nitrogen
బర్త్డే
converts nitrogen to ammonia.
హ్యాపీ బర్త్డే
See here, so this matter we can observe in this
మళ్ళీ పెట్టాలి.
image, the slide shows this is the nitrogenase enzyme.
ఏ చెప్తాను
Uses of this nitrogenase enzyme.
కాకుతే ఏది పడితే మెంటలోడా ఏది కాబట్టి ఇంత కొడతారు. ఎంత బరువు ఉంది
Is
దానికి చెప్పిపోయి ఇంత కొట్టి
this
కల
is
మీద
the nitrogenase enzyme.
మస్తు అది కూడా
Nitrogenase
పెచ్చింది.
and this is cell metabolism. And then after the mechanism, whenever the process is continuously progressive manner, then glycosylation of the dinitrogenase reductase is actively involved in this process. Okay, so in this process, see here, so nitrogen, dinitrogen is converted into ammonia. It is a usable form. So this is the dinitrogenase
ప్లీజ్
enzyme.
ప్లీజ్ చెప్పండి.
Same time, so
చూడు
the
చూడు
mechanism, dinitrogenase reductase mechanism we are observed is like.
ఏం కాదు పోతుంది. బరువు ఉంది అంతే అది.
So
దీన్ని కొడతా
this is the
తల
legume
మీదకి
plant. Have you ever seen
Charging
this legume
network.
plants?
ఇది కొడతా
ఏ
Root
charging
nodules. We
network.
are observing this root nodules, you know.
చెప్పేది అయితే ఇదే
Root
చెప్పు.
contains nodules.
అది ఉత్తే ఇష్టం ఎందుకు తీయాలి?
legume
మాకు అసలు
plants,
ఏది ఉంటుంది కొడతాడు.
they
చార్జింగ్
are-
పెట్టు
They
లేదు.
are having the root nodules or produce the root nodules
అవి
by the
కమ్యూనికేషన్ డాష్
involvement of the
ఎందుకు
rhizobium
ఇష్టం
bacteria
నాది
or microbial organisms. So the exchange of the nutrients during rhizobium legume symbiosis. See here, this is the symbiotic metabolic how to utilize both the association, the symbiotic association, how to involve to exchange the nutrients for survivalence process. So in this symbiotic nitrogen fixation process, this is the rhizobium legume association. This is one of the Fabaceae member or leguminaceae plant roots which we are observed in this image, right side. So these are the root nodules which is produced by bacteria. Okay, so bacteria are associated with certain plants families, mainly, primarily, especially legume plants. And the species make the largest single contribution to biological nitrogen fixation in the biosphere. See here, it is a pea plant can ability to sustain or produce number of root nodules with the help of rhizobium legume SRM. Okay, so in this inside the rhizobium, inside the root nodule, when we are cut down or break down or take a section, the root nodule inside we are clearly observed pink, light color pink or pink color, light pink color. We are observed. That is nothing but leg hemoglobin, a protein that carries oxygen to the bacteroids. Oxygen to the bacteroids. So that leg hemoglobin is acted as a oxygen scavenger. Oxygen scavenger. See here, it is a soybean plant. So soybean plant contains number of, we are observed number of nodules on the surface of the roots. Okay, so nodules develop by
అట్లా
interaction
నేను కొట్టిస్తా ఆగు.
broad rhizobium.
ఏ ఇస్తావు నువ్వు పిచ్చి వేషాలు వేయకురా,
One
నేను కట్టే
bacteria,
ఇస్తున్నా.
one bacteroids are the
ఎందుకు
what we
పడేయాలి
can say
నేను?
that root nodule pods we are
అది
observed in
తీసి
the
కష్టం అయితే అన్ని
root.
పడేసి పెడతాను
So one
మంచిగా.
root nodule we are observed. See here.
ఏ నాకు తీసిన కష్టం మీరు మంచిగా పడేసుకుంటారు. తీసిన కష్టం దగ్గర కట్టేది ఉంటుంది వద్దు ప్లీజ్.
And this is the nodule
కన్నయ్య
apex
చెప్తే
zone.
వింటాడు, I know that.
And this is O2 gradient zone.
కన్నయ్య
This is the
చెప్తే
O2
వింటాడు
gradient is
అనిపిస్తుంది.
there.
నీ బుక్ దే పోరా.
But inside, O2 is less
చెప్తే చేజాడిపోయి ఇప్పుడు మీరు
నేను రాస్తా.
రాసుకొని
just because of the leg hemoglobin
చేజాడిపోయి
absorbs the
ముందుగా మీరు హాస్టల్లో వేస్తే ఉంటది.
even though present
అది
of
ఇంకేదని
inside the
అంటారు, దాన్ని
nodule.
అంటారు
Okay.
కో ఏదో అంటారు.
Then see here.
పైన
This is here
ఎక్కించిన
the leg hemoglobin.
నాన్న
See here,
పైన
the
ఎక్కియడానికి
కోదండం.
nodule. See here.
మంచి
So
గుర్తు.
this is the
కోదండం ఎక్కించిన
external
ఉంటుంది.
aperture or
చేజాడి
external part of the
చూద్దాం
root.
చూస్తాను.
Nodule.
చేయరు
So this
స్కూల్లో
cortex
వాళ్ళు
of the
చూద్దాం.
root.
చూద్దాం.
At the same time, this is the
పైన ఇట్లా ఉంచుతారు.
light
క్లాస్
blue
పైన
color,
అందరూ మిమ్మల్ని
blue color
చూసుకుంటున్నా
region.
వచ్చి
They are
బ్యాడ్
belong to
బాయ్స్ అని.
vascular
చూద్దాం.
అట్లనే వినాలి
system.
క్లాసులు.
Maybe
చేయి ఇట్లా కట్టేస్తారు పైకి.
the
రాయమన్నారు.
complex tissue which we
వినమంటారు, అడుగుతారు, క్వశ్చన్లు
these are
అడుగుతారు.
present.
అప్
Vascular tissue.
కొంచెం అయినా ఉంటుంది. చూద్దాం.
The
చూద్దాం.
black color, that is the
నేను అన్ని
included.
మాటలే
జారిపోయింది మీరు.
So whereas
టీచర్ కూడా అంటాడు
inside
టీచర్ని.
the
వద్దు అది చెప్తే
ప్రిన్సిపల్ టీచర్ కి పోయి చెప్తాను నేను
we
కూడా.
are observed.
ఏమని
But the
గోడ
plant
మీద
cell
కలతో
is
కొడుతుంది.
a very
నేను స్కూల్
rapidly
మేనేజ్
plant cell is
చేస్తా.
going to
ఎవరు?
division.
మా టీచర్
Division
కూడా నేను
condition
చెప్తాను.
and newly
ఇప్పుడు ఏమని చెప్తా?
condition with low nitrogen fixing abilities. Here, the light
భయమే లేదు. అదే
brown or pinkish
చేయాలి
color region we are
నేను
observed.
చెప్తే
Then
వేయకుండా
mature
వినేసారా?
zone
మరీ
is
ఘోరం అయిపోయింది.
directly pink
బుక్
color zone
తెచ్చుకొని
we are
మీరు
observed.
ఏదైనా
High
ఇదే ఎట్లనే
nitrogen
చేస్తే
fixation
మనం వేసింది
ability
అప్పటి
which we are
ఇప్పుడు
observed
ఒక
in
రోజు
this
వేయాలి,
zone.
తినండి,
That is the
తినండి.
mature zone. Where at the
వేసింది
same
ఒక్క
time the
వేసిందా?
senescent zone is also there. So
ఒక
this
రోజు వేసిందా
senescent
బ్యాట్ పెట్టి?
zone, the
క్రికెట్
light
ఆడినాం
pink
కదా
is,
ఒక రోజు.
pink color is going to dark red or red color zone we are observed. That is the low zero nitrogen fixation ability we are observed. So this
అమ్మ ఆ రోజు
bacteria is
నువ్వు నన్ను
hydrophobic
బ్యాగ్
bacteria,
తీసుకొచ్చినావు కదా
even though they
సైకిల్
are
రోజు.
sorry,
అది
they
ప్లాన్
are
మొత్తం
autotrophic bacteria by because they are photosynthetically
ప్లాన్ వేసి
active.
మరి
They are prepared their own food material in presence of the sunlight. So these photosynthetic cells are
వాళ్ళు
here.
ఎట్లా ఎట్లా
So
ఉంటుండ్రో ఆటోమేటిక్లీ.
these nostoc or anabina cells,
Yes,
they
sorry.
have, even though the heterosis is also present. So
ఏం కాదు
the
టచ్ స్క్రీన్
heterosis
అయితే ఏం
cells
కాదు కదా.
is the specialized cells which get ability to fix the nitrogen from atmosphere. That the heterocyst is one of
ఇది
the
మనం
important cells of the trichome of
వాళ్ళు
the
ఇక్కడ
root vein
ఎట్లుండ్రు?
organelle members.
వాళ్ళే చేస్తారు,
So
వాళ్ళే
this is
క్లాస్
the
చెప్తున్నారు.
ఇది ఇక్కడ ఎట్లుండ్రు?
root nodule is
నేను
produced.
ఓపెన్ చేసిన కదా,
So the nodule
అందరినీ
formation,
జాయిన్
how to
చేసేది.
formation of the root
అట్లా
nodule.
కాదమ్మ,
అట్లా
బీచ్
చేస్తున్నారు.
ఎట్లుండ్రు
So
వాళ్ళు.
in this nodulation
teacher,
process,
వెనకాల
so these are the
వేస్తున్నారు
steps
అంతే.
we are observed. The first one is the,
అట్లా పెయింటింగ్
I mean, are you follow my dear friends?
అది వెనకాల
Are you
పెట్టుకోవచ్చు
feel good?
సెట్టింగ్స్ బ్యాక్ సైడ్.
Are you
అది
follow?
నాకు కూడా వచ్చి నాని ఫోన్ లో.
Okay. So the nodulation process,
అమ్మ మీకు
the chemical
చెట్ల గురించి
recognition of the
ఏమేం
root and
తెలుసు?
rhizobium,
చెట్టు
and the
గాలి
second
వస్తాయి,
step, that is the root hair
పచ్చగా
curl,
ఉంటాయి. చెట్ల
and
పండు వస్తాయి,
that
చెట్ల
root hair curl also we can call it shepherd crux,
సాల్ట
and formation of
సాయిల్
infection
అయిపోయి
threads,
సాయిల్ టాకర్
and fourth one is the invasion of the roots by rhizobia, and fifth one is the nodule tissue forms, and sixth one is the bacteria convert to bacteroids and begin to form nitrogenase enzyme, and lastly the seventh one legume provides rhizobia carbon, rhizobia, my dear friends, remember legume provides
బన్నీ
rhizobia with carbon.
కిన్నే
At the same time, Rhizobium provides the legume which gives you nitrogen. So nitrogen is provided by rhizobium and carbon is provided by legume plants. Are you follow or not?
ఎస్ సార్.
అమ్మా వినబడుతుందా?
Yes, sir.
లిగ్యూమ్ మొక్క అనేటువంటిది కార్బన్ అందిస్తుంది. కార్బన్ కంటెంట్ ను బ్యాక్టీరియాకి అందించేది లెగ్యూమ్ ప్లాంట్స్. బ్యాక్టీరియా ఏం చేస్తుంది లెగ్యూమ్ ప్లాంట్ కు? నైట్రోజన్ అందిస్తుంది. సో దిస్ association organism get benefited. So this type of association we can call as symbiotic association. Okay, so this chemical recognition of the roots and rhizobia, root hair curling, formation of the infection threads, infection of root by rhizobia. So these are the 7 steps we are observing. Okay, so this is today's class, my dear friends.
ఏది నాకు ఏం 19 అంటే 7 అయిపోయిందా?
Okay, so these are the how root nodules are produced on the surface of the root.
అన్నందుకు అమ్మ రోజు తొందరగా లేట్ వస్తావు కదా.
9 మీతో
My dear friends, I will stop here. If you have any doubt, please ask me, my dear friends.
మై డియర్ ఫ్రెండ్స్ అతను అడుగు ఏమన్నా డౌట్స్ ఉంటాయి అడుగు.
No doubt.
No, sir. Thank you,
ఆ
sir.
డౌట్స్
So
ఉన్నాయి.
see here at the same time, some of the legumes they are belongs to other than bacterial members or other than legume plants also involved. See here, alpha not belongs to legumes, you know. So clover, soybean, beans, peas, sesbania. So these are all host plants. So bacterial symbiote is different. Rhizobium meritoli, Rhizobium trifoli, and then Bradyrhizobium japonicum, Rhizobium caesioli. So Rhizobium leguminosarum, Azorhizobium, Colonodons. So these are the other bacterial symbiotes which they are involved in different plants. They are belongs to Leguminaceae family members. Okay. Right? So I will stop here, my dear friends. Tomorrow we will going to discuss about the other new topic. I hope you clearly understood
మీ
today's
సార్
topic.
పేరు ఏంటి అమ్మ? తిరుపతి రావు సైనింగ్ ఆఫ్. బై బై.
ఏమైనా doubt ఉంటే అడగండి,
Time
my
for
dear
math
friends.
and
ఏమైనా doubt ఉంటే అడగండి. Any doubts?
No doubt, sir.
No doubts? Okay.
నువ్వు కొద్దిగా
Tomorrow we will meet. Okay, my dear friends, I will stop here. Madam ji, Manjula Madam ji.
ఎస్ సార్.
Okay, thank you very much.
कभी याद करे तो तुम्हारा.
Madam,
ఏ మాట్లాడకు. ఓకే సార్, థ్యాంక్ యూ సార్.
right. Okay, with your permission, I leave today class.
ఎస్ సార్, ఎస్ సార్.